Everyone, and welcome. I am happy to host the webinar today, presented by Dr. Sharmista Pal and Jayashire Raju Borad. The webinar today is determining minimum effective polymer concentration for amorphous solid dispersions using Crystal16. This webinar is made possible today with the help of Spincotech. Spinco is our colleagues, our partners in India, a company with 45 years of legacy, true to bringing technology and enabling science. They have more than 60 support centers all over India, delivering solutions across chromatography, mass spectroscopy, life sciences, process technology, and lab essentials. And of course, they are distributing and helping our customers in India with Technobis instruments. Just before I start, I would like to tell you that this webinar is being recorded and is made available in a few days. You will receive an email where you can rewatch the recording or download the slides of this presentation. If you have questions, we will take them at the end. Please make use of the Q&A section of this platform, and you'll be able there to write your question and we will answer them at the end. If you still have questions after the webinar, please contact us at info@crystallizationsystems.com and we will do our best to share the questions with our presenters today, or we will try to address ourselves these questions. Just before I start, I would like to share a bit of information about Technobis. Who is Technobis? We are a Dutch company located in the north of the Netherlands. We are a world-leading technology provider for solid-state research, process development, and formulation. We have two product portfolios, one on crystallization systems, crystallization screening. Also, our webinar today, you will see a few case studies related to this portfolio with three products in the portfolio: Crystal Breeder, Crystal16, and the Crystal line, and a more recent reaction screening product line with the React-All. The React-All, reaction screening product line is a multiple reactor system with 5 reactors working in parallel. As you can see it here, the beauty of it is the automated sampling. Once you perform your experiment, the sample can be drawn at the moment that you would like to, immediately is quenched, diluted, and transferred to an HPLC vial, LCMS vial that you can further analyze. It's robust enough for heterogeneous and homogeneous reactions. So, as I mentioned, route scouting, reaction screening, optimization, kinetics, everything you can do with the React-All. Now, the crystallization product line, the Crystal Breeder, is your instrument that works at microliter scale, is dedicated for growing single crystals, solvent screening, some very early stage solubility determination. It comes with gadgets, as I like to call them, evaporation, vapor diffusion. Quite a lot of crystallization methods are available on this product. The Crystal16, and some of you might know the Crystal16 and the Crystal line. If I look at the names of this webinar, the Crystal16 is the instrument for solubility, metastable zone width, solid state screening, you name it. You are able to determine temperature, determine solubility data. So it gives quite a lot in a very short time. The Crystal line with the camera technology that really works like a microscope, 8 independent reactors, each one of them comes with cameras, or Raman helps. If you have a Raman spectrometer in your lab, you can connect it and you can perform any type of development, optimization, process development, crystallization, formulation, process for optimization type of experiments with it. It comes also with an AI-based image analysis that allows you to do particle size distribution, size, shape, particle count, quite versatile. It goes in magnification camera, goes resolution as low as 1-2 microns. Just to recap, Technovis manufactures lab instruments and brought to India by our colleagues from Spinco, which enables you to measure, characterize, and control some of the parameter properties related to crystallization, which you will hear a bit more about this today. Now, I am happy to introduce to you our speakers today. The first speaker is Dr. Sharmista Pal. She is the solid form screening scientist at Dr. Reddy's laboratories. Actually, she is leading the solid form screening department at Dr. Reddy's in India. She has more than 20 years of experience in the pharmaceutical industry, working in area of solid form development, manufacturing, and characterization. Her expertise is in the field of developing and modulating physical, chemical, and bulk properties of API through solid form transformations and crystallization processes to address challenges in formulation and bioavailability with regulatory framework. Prior to Dr. Reddy's, she worked in different scientific roles at big leading pharma companies and CROs, big pharma like Eli Lilly, AstraZeneca India, Biocon BMS Research Center. She holds a PhD in pharmaceutics from University of Minnesota. And our second presenter today is Jayshree Raju Borad. She is a polymorph scientist, at Dr. Reddy's, working at Dr. Reddy's for about 2.5 years experience in pharma industry, holds a master degree in pharmaceutics from Naipur Mohali, and actively contributes to advancing solid form understanding in drug development. And with this, I would like to thank our presenters today. I will stop sharing. Sharmita, thank you very much for being available and sharing your experience and case studies from Dr. Reddy's with our viewers today. Sharmita, the floor is yours. Please, you can start.
Yeah, thanks, Carmen, for giving us the opportunity to present our work that we have done on determining minimum effective polymer concentration using Crystal16 for ASDs. Me and my colleague Jayshree will be presenting this webinar today. So before we start, a little bit about our company that we work in. Dr. Reddy's is a global biopharmaceutical company headquartered in Hyderabad, in India, and the operations spread over 74 countries with more than 20 manufacturing sites complying to GMP facilities. And our motto is "Good health can't wait," and we strive to achieve it through affordable and innovative medicines worldwide. So now coming to the topic of work today, topic of discussion today is on amorphous solid dispersions. So we will start with the importance of amorphous solid dispersion in pharmaceuticals. So as we know that the drug substances or the APIs, they are classified into 4 bioclassification system basis their solubility and permeability. And a molecule which has high solubility and high permeability is expected to have high bioavailability, whereas as the solubility or permeability goes lower, then there are there may be issues with the bioavailability. And that's the reason this bioavailability classification system has been developed to assess the risk of developing molecules. So in the left-hand slide, on the left-hand side of the slide, you can see the cases of marketed APIs in the past, where class 2 and class 4 combined were about 50% of molecules. And these constitute compounds where the solubility is very low, and that becomes the deterrent for adequate bioavailability. In the coming future, the expected drugs, the drugs that are coming in the pipeline are expected to be even have more solubility problems. So as you can see here, the percentage of class 2 and class 4 drugs are now increasing to almost 85 to 90%, which will create a problem, anticipated problem in providing adequate bioavailability. There are several mechanisms or techniques that can be utilized to improve the bioavailability, which occurs because of low solubility, such as by modified solid forms like polymorphs, co-crystals, or by changing the physical nature of the material by particle size reduction, increasing surface area, and things like that. So one of the other technology is making the API as a amorphous solid dispersion. So in amorphous solid dispersion, what is done is the API is, which may be crystalline or which could be amorphous. It is mixed with the polymer to make a homogeneous mixture or a homogeneous dispersion. The advantages are that the molecules are of the API, they are far apart, and at the same time, the polymers create a barrier for the molecules of the API to come together during shelf life, and hence retaining this homogeneous dispersed structure for a longer duration. We will come to how it actually helps in improving solubility and bioavailability in a little bit. So the polymers which are usually used to make ASDs can be classified on the basis of their chemical structure or on the basis of their ionic nature, as has been shown here. So mostly in the pharmaceutical organizations, it's the cellulosic or the vinyl pyrrolidones and their the other amphiphilic molecule polymers, they are used for making the ASDs. And some of these cellulosic can also be ionic, or they can also be non-ionic polymers. So now coming to why amorphous solid dispersion is looked at, especially compared to the possibility of making other solid forms like polymorphs or co-crystals or amorphous, is that first thing, one of the primary reasons why crystalline forms have low solubility is because of their high lattice energy. Now, breaking that lattice energy by making the API amorphous actually increases the solubility. So that's what you can see as the spring effect of amorphous material itself. However, the disadvantage can be with amorphous material is that because of its higher solubility, which is a good thing, but because once it dissolves, it creates a high supersaturation, it can immediately precipitate or crash out. And if it does so, the solubility can just come down to the crystalline form itself, rendering the advantage due to the amorphous null and void. So essentially what we want is to not only get the high solubility, but also sustain the high solubility for a longer duration, that it can be absorbed properly during the drug stay in the gastrointestinal tract. So amorphous solid dispersion can help in sustaining the supersaturation in addition to increasing the solubility. So that's the advantage of amorphous solid dispersion from the solubility perspective. Also, sometimes the solubility of the amorphous itself may not be as high as we require, and the presence of the polymer enhances the solubility beyond the solubility of the amorphous. So in these cases, the polymer act not only to sustain the supersaturation once the amorphous solid dispersion is in the media, but it actually also helps in increasing the solubility more than that of the amorphous. So with that now covered, let's move on to the role of polymers in the preparation of ASDs. So as you can see on the left panel, when different polymers are used for making an ASD, different polymers can have different effects on the extent to which it increases the solubility of the API. So as you can see here, as a schematic, that from polymer 1 to polymer 6, the solubility increase decreases quite a bit. The enhancement of the solubility decreases quite a bit. So this actually helps in identifying which polymer may be more suitable to obtain the supersaturation or the higher solubility that we are looking for. Now, even for the same polymer, the solubility effects can be impacted by the percentage of that polymer in ratio to the API present in the amorphous solid dispersion or ASD. So in the right panel, as you can see, for the same polymer, if different amount of, for it is the same polymer, but different amounts of the polymer have been added to make the ASD, as the percentage of API increases, which means as the percentage of polymer decreases, there is a certain threshold beyond which you see there is a sudden drop in the dissolved percentage of the API. And this region or threshold where there is this sudden change in the solubility occurring is called the LOC, or the limit of congruency. There has been a lot of studies on why this might be happening. We will not go into that detail because our presentation is more towards finding this limit of congruency in a faster manner. The reason being, what is the importance of this LOC is that if the polymer concentration is below the LOC, as we can see, the enhancement of the solubility of the API is really not that much. The enhancement of the solubility of the API really picks up after or when the polymer concentration is more than the LOC. So as we will be presenting later, so it is right now understood that if the polymer concentration is above the LOC limit, or the more content of polymer is available, then the solubility of the sustenance of the solubility and the solubility of the API will be higher, much higher than that of the API, whereas if the concentration of the polymer is lower than the LOC, there could be chances of low solubility itself, as well as chances of immediate precipitation of the API. So now what we have intended to do in this presentation, as I said, is to show you an alternate way of how we can get to the LOC or limit of congruency. But first, I also want to share with you the traditional way, which will help you to appreciate how the new way of getting the LOC, or as Jayashree will be talking about MEPC, how that can be helpful. So the traditional way of obtaining the limit of congruency or the minimal polymer concentration to sustain the supersaturation or the critical polymer content is that first we disperse the ASD in the media, and these are ASDs prepared with the same polymer system with different concentrations or compositions of the polymer. Then withdraw samples as a slurry at timed intervals, centrifuge to collect the mother liquor, and then the mother liquor is analyzed by HPLC or other proper technique to get the concentration of both the API as well as the polymer. So this is, as you can see, that this and the way the data is analyzed is that for these different timed intervals, the concentration of the API and the polymer, they are plotted together. And as we can see, when this is plotted for the different ASD compositions, there is a particular composition or a range of composition where we start to see a drastic change from proportional dissolution of the API and polymer to a disproportionate dissolution of the API and polymer. So you can see the proportional dissolution of the API and polymer on the left panel, and on the right panel, it's a disproportionate release of the API faster than that of the polymer. Sorry, the other way, the API dissolves faster than that of the polymer. So this creates this identification of this polymer concentration where this happens. As you can see from the traditional way, it can take quite some time because we have to first prepare the ASD, then prepare the slurries of those different ASDs. Material requirement is higher, and each of the samples then have to be analyzed by HPLC or other optimal analytical method to get the API and polymer concentration. So the alternate technique, which my colleague Jayashree will be talking about, that can be much shorter, and I will hand it over to her for to go over the procedure with you. Thank you.
Thank you, ma'am, for explaining the fundamental concepts of supersaturation and the limit of congruency related to the amorphous solid dispersion. Building on that, I will now take you through the experimental approach and how we determine the critical concentration of polymer using the Crystal16. As we know, Crystal16 is a high throughput crystallization platform, and it enables simultaneous monitoring of multiple samples under the controlled conditions. The system monitors samples in real time using transmissivity or turbidity measurements, so we can clearly observe when crystallization begins. One key output from the Crystal16 is the induction time, which tells us about the onset of crystallization. Go to next slide.
Yes. This is,
yes. So moving further, this slide represents the overall experimental design used in this study. So the main objective is to determine the minimum effective polymer concentration for ASDs by Crystal16, and the study is carried out using the solvent shift method, where supersaturation is generated by introducing the API solution into an anti-solvent containing polymer. Coming to the materials, the model compound used as MC1 and the polymers evaluated are copovidone, HPMC, and PVA. Methanol is used as the solvent for API phase, while pH 1.2 chloride buffer acts as the polymer phase. The API stock preparation is as shown in the figure. So let's see one by one. So here we begin by selecting stock concentration of 15 mg per ml. This is intentionally kept well below the saturation limit to ensure complete dissolution and to avoid any precipitation, which is critical for the accurate dosing. Next, we accurately weigh the required quantity of API, referred to here as MC1, and transfer it carefully into a vial. So to dissolve the API, we added 1 ml of methanol, which acts as the solvent. The vial is then subjected to the sonication, which helps in achieving a completely clear and homogeneous solution. This step ensures there are no undissolved particles that could affect the experimental reproducibility. And at this stage, our final stock solution concentration remains 15 mg per ml. Now, from this stock solution, we took an injection volume of 20 microliter for each experiment. And based on this, the amount of API introduced per experiment is 300 microgram. So overall, this method ensures a precise, reproducible, and fully dissolved API stock solution, which is essential for a reliable analysis. Please move to the next slide.
Yes.
Yes. Now let's move on to the polymer stock solution preparation, which plays an important role in the system. So first, here we began with polymer solubility testing using a pH 1.2 chloride buffer. The polymers used here include copovidone, HPMC, and PVA, as shown in the slide. Each polymer is tested to ensure it completely dissolves in the buffer, forming a clear solution. Next, accurately weigh the 100 mg of selected polymer. So make sure precision at this step is very important to maintain the final concentration. In the third step, we gradually add the pH 1.2 chloride buffer to the polymer while continuous stirring. And this stirring ensures uniform mixing and prevents the lump formation. Then we adjust the total volume to the 10 ml, which gives us the desired final concentration of 10 mg per ml. After the preparation of solution, that solution is filtered using the 0.45 micron filter to remove any undissolved particles. And finally, we obtain a clear particle-free polymer solution, which is ready for the experimental use. Go to the next slide.
Yes.
Yeah. Going forward, this slide shows the stepwise workflow of Crystal16 program used to evaluate the nucleation behavior and determination of induction time. So we started with the step 1, preparation of polymer solution in vials, where polymer stock solution added, followed by pH 1.2 chloride buffer to maintain a fixed total volume, which was 0.9 ml, and each vial represents a different API to polymer ratio. In step 2, which is vial loading, all prepared vials are placed carefully into the Crystal16 instrument, ensuring proper sealing and correct positioning for the uniform experimental conditions. Next is step 3, pre-equilibration, where the system is stabilized at 25 degrees Celsius for 5 minutes. And this step is critical because it ensures that all the vials starts at the same thermal conditions. Now, once the equilibration completed, the instrument provides a prompt for API injection. And this ensures that the experiment begins only after achieving the thermal stability and minimizing the variability. Moving to the fourth step, API injection. A fixed volume of API solution is injected into each vial, and this initiates the solvent shift and creating the supersaturation. In step 5, which is mixing and stirring, continuous agitation ensures the uniform distribution and consistent supersaturation throughout the experiment. Sorry, just go back. Yeah.
Yeah, I think is timing on each one of the slides, so that's why it goes. I don't touch anything, so that's why. But it's okay. We handle it. Go forward.
Okay. Yeah. So lastwise, step 6, which is a real-time monitoring. So this is the most critical part of the experiment, and in this, Crystal16 instrument continuously provides the real-time data on turbidity or percent transmissivity of each vial over the entire duration of the experiment, typically up to the 24 hours in this particular experiment. So initially, you can see in the graph, initially when the solution is clear, the instrument records high transmission value. As the system reaches supersaturation and nucleation begins, the particle starts forming, which leading to an increase in turbidity and corresponding drop in the transmission. And this change is captured as a function of time, allowing identification of the exact point where turbidity starts, known as the induction time, or the point is known as the nucleation point. So we can say that the measured induction time reflects the system supersaturation levels, and this relationship helps us to define the LOC for the given API polymer system. Additionally, saying the instrument enables the comparison across different API polymer ratios by tracking how long each system maintains clarity, thereby giving the insights into the polymer effectiveness in delaying the crystallization. So overall, the Crystal16 provides the continuous and accurate kinetic information without disturbing the system. Please go to the next slide.
Oh.
Yeah.
Yes.
Last one. 12 number.
This.
Just go back one slide. Go back, 12.
I'm on the 12th.
Yes, yeah, yes, thank you. The output graphs obtained from the Crystal16 at the end of the experiments are shown in this slide. Let's understand what exactly it represents. So in this graph, the x-axis, let's focus on figure number 1. So in the graph, the x-axis represents time in minute, while the left y-axis represents temperature in degrees Celsius, and the right y-axis represents the percent transmission. You can see the red line indicates the temperature which is maintained constant throughout the experiment to ensure the controlled conditions. The light blue line represents percent transmission, which reflects the clarity of solution and is directly linked to the crystal formation. This is the most important curve. Initially, we can see the line is flat, indicating the solution is clear and the transmission is high or stable, and no crystal formation. And as the crystallization begins, the transmission sharply drops because the solution becomes cloudy due to the crystal formation. And this point is considered as the cloud point, and the time is called as the induction time, which indicates the onset of nucleation. The vertical lines in the graph represents crystallization events occurring in individual vials at different time points. Each vial crystallizing at different time points. Simply saying, these vertical lines basically represents where crystallization starts in different vials. Therefore, the Crystal16 profile help us to evaluate the induction time, nucleation behavior, supersaturation stability, and for movement of the system towards the LOC region. So now let me take you all through some real examples that we perform in our lab using the Crystal16. So the first example is with the copovidone as polymer. And these graph shows these all graphs shows the Crystal16 analysis of the model compound MC1 with increasing copovidone concentration. So basically, these are the real-time transmission profiles obtained by the Crystal16, starting from the 0 polymer concentration and progressing to the 1:0.25 API is to copovidone concentration to 1:9. So just move to the next slide.
This one? Oh, no.
Yeah, next slide.
This one.
Yeah, thank you. So, no, next.
Next one, sorry. This.
Yeah, yes, thank you. So, let's focus on the graph, which is induction time versus API is to polymer ratio graph. So we can see there, without polymer means at a 0 polymer concentration means API only, and at the lower ratios like 1:0.25 and 1:0.5, there is an yearly drop in transmission observed with very short induction time, which kind of indicating the rapid crystallization and poor supersaturation stability. This means the system still far from the LOC region. As the copovidone concentration increases to the intermediate ratios such as 1:1 and 1:2, the crystallization is delayed, but showing the partial stabilization of supersaturated state, and still polymer concentration is not sufficient. But if we see at the ratio 1:3, a significant increase in the induction time of around 24 minutes, which we can see in the graph, indicating strong nucleation inhibition and effective supersaturation stabilization. This suggests the movement of system closer to the LOC boundary where nucleation becomes controlled. Although the higher ratios like 1:4, 1:7 and 1:9 showed even higher induction time, but still they are not selected as the MEPC because MEPC is defined as the minimum effective polymer concentration required to stabilize the supersaturation. But at 1:3, there is a threshold behavior with significant increase in the induction time, indicating the effective nucleation inhibition. But beyond this point, beyond 1:3, further increase in hello?
Yes.
Yeah. Further increase in polymer load leads to diminishing returns and the formulation constraint like reduced drug loading, and hence 1:3 selected as the MEPC, where the system maintained at near the LOC boundaries for the copovidone. Go to next slide. Yeah, coming to the second example, which is with HPMC as a polymer. So HPMC as a polymer, and again these are the real-time transmission profile obtained from the Crystal16 for the HPMC with the different concentrations. Go to the next slide, please. Yeah. Okay. Thank you. So here also, next slide.
16, right?
Yeah, 17.
Okay. Yep.
Okay, thank you. So let's focus on the graph. So we observe, so before we observe a trend, so with HPMC also, we observe a trend similar to the copovidone. So as we can see that up to the 1:2 API to HPMC ratio, only moderate increase in the induction time is observed. But at the 1:3 API to HPMC ratio, there is a sharp jump in the induction time, which is at 80 minutes, and this represents the threshold behavior. Below this concentration, polymer is insufficient, and at this point, 1:3, the system transitions into the strong stabilization near LOC. So this basically indicates a critical concentration required for the effective nucleation suppression. So if we move beyond the 1:3, at 1:4 and 1:9, although the stability is maintained, but the improvement not proportionately significant. So 1:3 is identified as the MEPC, which is minimum effective polymer concentration for HPMC, as it effectively maintained the supersaturation and delayed the crystallization, while higher concentration only showed the diminishing returns without meaningful shift in the LOC. Please go to the next slide.
18, right?
Yeah, yes, thank you. So now looking at the last example, which is with PVA, polyvinyl alcohol as a polymer. So again, these are the real-time transmission profiles obtained from the Crystal16 at the different APIs to PVA ratios from higher to lower. Now go to the next slide.
Yes. Oh, this one?
20. Yeah, thank you. So with PVA, we observe a different behavior compared to the other two polymers. So in case of PVA, we can see that, just focus on the graph. So we can see without polymer, crystallization occurs quickly with a short induction time, which indicating rapid nucleation from the supersaturated state. At lower ratios like 1:0.25 and 1:0.5, so there is some stabilization with a slight increase in the induction time. However, as we move further, increase in the concentration of polymer from 1:1 to the 1:9, we saw the opposite trend where induction time actually decreases and the crystallization occurs earlier. So this suggests that the higher PVA concentration reduces effective supersaturation, likely due to decrease in solubility or unfavorable or nonlinear drug polymer interactions, or flipping from the nucleation inhibitor to the nucleation promoter at the threshold concentration. So as a result, the system exits the supersaturated state faster and promoting earlier nucleation and then crystallization. Talking from the LOC perspective, in case of PVA, instead of expanding the metastable zone, the higher concentration of PVA shifts the system towards the faster crystallization, which narrowed the supersaturation window. So therefore, identifying the both upper and the lower concentration limits become critical in case of PVA. So we can say that the PVA exhibits the concentration dependent dual behavior. Go to the next slide, please. Yeah. Coming to the comparison between all three polymers, copovidone versus HPMC versus PVA. So copovidone and HPMC shows a typical behavior where increasing the concentration increases the induction time and the stabilization supersaturation with the minimum effective polymer concentration MEPC is at around 1:3. In contrast to that, PVA shows opposite behavior. Beyond 1:0.5 API to PVA ratio, increasing the concentration reduces the solubility and induction time, indicating a shift from nucleation inhibition to the nucleation promotion. So overall, copovidone and HPMC, we can say, are the effective crystallization inhibitors, while PVA shows the concentration dependent dual behavior, making 1:0.5 as its optimal MEPC, but there may be an upper and lower limit which needs to investigate or study further. Let's go to the next slide. Now coming to the key takeaways from this study are, first, we can say that induction time proves to be a reliable indicator of nucleation delay and overall supersaturation stability, helping us to understand crystallization behavior more effectively. Second, solvent shift method successfully generates the supersaturation, making it suitable and practical approach for studying the crystallization process. Third, the polymer addition. The polymer addition plays a critical role, especially in case of copovidone and HPMC. Polymers like copovidone and HPMC help to sustain the supersaturation and significantly delay the crystallization. However, we also observe that certain polymers, like here example is PVA, as we saw, the behavior can be counterintuitive, suggesting there may be optimal concentration limits that requires further investigation. And finally, the MEPC, minimum effective polymer concentration, provides lowest concentration of polymer, showing the significant nucleation delay, not highest concentration. Next slide, please. Hello?
Yes, it is there.
No, one more slide is there.
No?
Yeah, thank you. Yeah. So the last but not the least, this slide summarizes the key experimental advantages using the Crystal16 system. First, low material consumption. So for performing this experiment, we require only about 15 mg of API, around 100 mg of polymer and less than 20 ml of solvent. And this is extremely beneficial in early stage development where material availability is limited. Second, rapid turnaround time. So with Crystal16, we could generate the meaningful supersaturation and crystallization data within just 24 hours, and this significantly accelerates our screening process. Third, efficient screening. The system allows simultaneous evaluation of multiple polymer types and multiple concentrations at a single time. So this helps us quickly to understand the polymer effectiveness and concentration dependence in a controlled and reproducible way. And finally, the time and resource saving. So by automating and parallelizing the experiments, Crystal16 minimizes. Can you please? Okay, please. Yeah. So Crystal16 minimizes the manual efforts while enabling the faster and data-driven decision making in ASD development. So overall, using the Crystal16 makes the entire screening process faster and more efficient. Thank you.
Oh, wait, this was the last one, right?
Yeah.
I'm really sorry. I was not touching anything. It was just going by itself on different slides. So I could not really control it.
It's totally fine. Yeah, it's totally fine.
Thank you very much both for presenting this topic on amorphous solid dispersions. Just one second, I'm trying to see now. We do have a few questions. I will try to read to you. I hope Sharmita is still there. So one of the questions is, if you want to conduct solubility at below 1 degree for sensitive API, how should I detect solubility using Crystal16? Well,
sorry, can you repeat?
Yeah, so if I want to conduct solubility at below 1 degree, so at below 1 degree would be 0 minus or so degree C. For a sensitive API, somebody is asking, how should I detect solubility using the Crystal16? Can I step in?
Sensitive to what? Like sensitive to heat?
It could be that the customer is asking for temperature. Indeed, I take this one, Jay. Some compounds they behave differently. Like for example, well-known also the polymers. Some polymers they behave like this, but also like the calcium carbonate in the shower. When you put high temperature, it crystallizes, and when you have low temperature, is dissolved. So it could be that maybe the customer or the viewer is asking for those cases. It's still the same. You look, it doesn't matter the temperature that you are setting. Of course, if you look at lower temperatures, go ahead and look at you can use with the Crystal16, you can go to minus 25 degrees by the use of a chiller. And the same, you look for the clear and the cloud point. So because you are looking for solubility, you need to look for the clear point and avoid the cloud points. It doesn't matter what system you have. The same idea of clear and cloud point, no matter the temperature, you should be looked for. Somebody is asking for a figure 1 system, but now I do not know which figure 1 system. She said, maintains same temperature throughout, then how nucleation and cloud point determined. Let me try to go back to a figure 1, Jay. Hold on with me.
Yeah.
Here is maybe the figure 1. What do we see here? You try the
actually in whole experiments we have maintained the constant temperature, like whether it is a figure 1 or whether it is a figure 4, doesn't matter.
Yeah.
For all we maintain the temperature 25 degrees Celsius.
Yes.
to
Jay, I don't hear you.
hello.
Yes, now I hear you. Hello?
So here,
Yeah,
hello,
yeah.
hello.
Yes, Jay, I hear you.
1 second, 1 second.
Jay, I can hear you.
Hello, hello, hello.
I hear
So
you.
here,
Yeah?
actually we wanted to, yeah, hello.
Yes.
So we here, by keeping the temperature at 25, we tried to mimic the in vivo environment first. Second, we chose the temperature, control temperature to for determine the induction time is mainly due to the polymer concentration, not for what to say, temperature variation. So here we change the concentration of polymer by keeping the temperature constant, which mimic the in vivo conditions.
Yeah. Another question, Jay. What is LOC?
LOC meaning the limit of congruency.
Okay.
So it basically refers to the point or the boundary where the supersaturation solution becomes unstable and the crystallization or nucleation starts occurring rapidly. So we can say, like, before LOC, solution will be relatively stable. Near LOC, nucleation risk is increases, and beyond LOC, the rapid crystallization occurs. So
Okay. Jay, one more. We have a few, so hold on with me. In what kind of APIs crystallization inhibitions helps? Is it in drug product, such as type 4? I guess class 4 is trying to say.
yeah.
What
Sorry,
type of, what type of APIs you have used for this? Where you were successful to inhibit crystallization for them?
So here we can say the formulation is amorphous solid dispersion.
Yes.
So if we compare the crystalline and amorphous system, so
Yeah.
amorphous system is having the higher solubility,
Yeah.
right? And the polymer is the if we suppose the drug is, if even though our drug is crystalline, which will having the low solubility, the polymer will interact with that crystalline API, forming the polymer matrix and
Yes.
hold. Sorry.
Yes, yes, yes. Just that the customer is asking what type of drug. It was drug product or drug substance?
It was API. It is a drug substance.
Drug substance. And can you say
Yes.
anything about the classification? What class was? What type of class? Is it a problematic one to? Do you remember that, Sharmita, in the beginning?
Basically, class 2 and class 4, because
Okay.
class 2 and class 4 of the BCS class having the less solubility, and because of that less solubility reason, we are approaching to the amorphous solid dispersion as a strategy. So yeah.
Okay. Is any scientific paper with these studies that you have published, maybe?
Not yet.
Not yet. Okay. Somebody else is saying, I think this experiment time and concentration is based on the maximum daily dose API polymer with at least 4 hours time. Makes this sense?
Sorry, I didn't get.
So somebody is
Can
saying,
you please?
Somebody is saying that the experiment time and concentration used is based on the maxi daily dose of API ratio polymer. Is this right? Were you trying? How
Carmen,
much? Yeah.
am I audible?
Yes.
Yeah, Jayashree, if you can just mute yourself. Yes, ma'am. Yeah. Can I answer this?
Yes, please.
Yeah, maybe Jayashree, you have to log out of Teams maybe for just a minute.
No. She doesn't have to. We can hear you well, both, so it's okay.
Okay. So the question is whether it is the time period
Yes.
of the nucleation and the dose concentration that we determined.
Yes.
That was with respect to the maximum daily dose or limit.
Exactly. So this is exactly what the attendee was asking.
Yes. So the answer actually, is that while we started with, we strive to do the maximum daily dose and kind of normalize it to the experimental concentration. However, the results that we were seeing were not. It was so dilute that the result was not coming. So we had to optimize on the concentration to see the effect of the polymer concentration on the LOC. But just to let you know what we have found, which we cannot actually talk about or publish, we can talk about it but can't publish, is that when we have compared it against the actual LOCs determined by the more traditional way, they are much more comparable.
Okay. Somebody else is saying something. Is it helpful in screening the polymer to look also for high drug loading? So if you do such a polymer amorphous solid dispersions, is it useful also to consider high drug loadings or amorphous solid dispersions? I think you should be conducted by the daily dose.
Answer that? Hello?
Yes. We can hear you, Jay.
Yeah.
So
just to answer, yes, definitely we can also look at high drug loading.
Yeah.
The purpose here was to actually see what is the minimum polymer required, because in most cases what happens is if the polymer concentration is below this threshold, then the ASD is not stable at all. So we need the minimum polymer concentration, otherwise we will have to do a lot of experimentations to identify what amount of minimum polymer we should be adding. So if these experiments indicate that the LOC is pretty high from the respect of the API concentration, we can definitely reduce the polymer concentration and it can be a high drug load API ASD.
I have another question related to the buffers. You have used buffers maybe between 1.2 and 6.8. How would you choose the buffers? How do you choose the buffers for experimentation? Sharmita, can you answer that?
Yeah, so the main reason was that these are the main physiological pHs at which the solubilities are looked at for ASDs. So, I mean, these are very restrictive buffers that you can see, right? Because under the full analysis, we have to look at different pH buffer medias. So what we have presented today is only pH 1.2 and 6.8, but definitely it can be done and has been done for other pH conditions also. So yeah, it is basically this buffer simulates the gastric conditions which are in our body. So that's why these buffers they have utilized.
Another question, can we evaluate the surfactant effect in delaying nucleation in supersaturation for screening suitable surfactant selection? I think that would require some modeling. I haven't seen this in your studies. Have you done such a study?
Yes, so we have done predictions on the surfactants of how it might impact the stability as well as solubility. But I think you are asking from the experimental perspective.
Yeah.
So yeah, we have thought about it, but we haven't really done it for this particular model system.
What if?
Yeah, that is definitely a good thing to try out.
Yeah, yes. that is one of the good area to explore. We'll definitely try it.
Yeah. What if the material is too fibrous or hard to manage at micro level to weigh in and to dissolve? What would be your advice? So I guess hard to dissolve at microliter level would be that it's very unsoluble, and then also too fibrous.
Right. So actually, this is where we think that, you know, this procedure can be pretty advantageous, because here we are not really dealing with solid API to be placed in each vial. We are making a stock solution of the API, and that can be made with a larger quantity of the API if it's a handling issue. And then it can be diluted according to the concentration that we need. So because here minimal quantity is not, for each vial, measuring the minimal quantity of the API in the solid form is not required. Hence, it might become easier to do the experiment.
Yeah. And somebody is also asking if it's possible to do micro level at Crystal16. Yes, that question I will take. It's still possible. We go slow with the transmissivity. We can see a few microns and very low concentrations. So indeed, if you are going towards the nano particles, and so it would be a bit more difficult. Just one more question. Jayshri, thank you for your detailed experimentation presentation. What was the starting point of induction point? Was it start of the experiment or after specific saturation reached?
So as I mentioned, so first equilibration is needed. So which was for 5 minutes, which was just with polymer. And when I charged my API solution, so that time, the first drop in the transmission, and that time point was the induction time we considered.
Somebody else is asking how to go beyond LOC with stable ASD, so amorphous solid dispersions. Is it possible to tackle the LOC with ASD?
Ma'am, will you please answer? Hello? Hello?
Yes?
Sorry, I didn't actually get the question. Can you please repeat?
Yes, how to go beyond LOC with stable amorphous solid dispersions. Is it possible to tackle the LOC?
Okay, I understood the first part, so I'll try to answer that, but I couldn't understand the last sentence. So the question is regarding, can we go beyond the LOC?
Yes.
So LOC is very system specific, right? So this LOC will vary because of the kind of polymer which is used, as Jayashi showed in her work, right?
Yeah.
So if we have that freedom to change the polymer, we can go to a higher drug loading. Now, if we want to stay with the same polymer,
Yeah.
we can also change the surfactant to move the LOC. So that's another way we can probably manipulate.
Yeah.
So those would be the strategies I would say. And there are combinations of polymers that can also be used, because anything which changes the system can potentially change the LOC, and it does.
Is there any relation to glass formation ability? So the glass formation, can we determine induction time for glass formation ability class 1 and 2 as well? So is any relationship with the glass formation?
Yeah, actually, that's a good question. We haven't actually evaluated that, but maybe that will be something good to see, that for the different glass forming ability type of molecules, does it provide the same kind of assessment or not? But we haven't done this as part of this study.
Okay. Somebody is asking another question not related to ASD, to amorphous solid dispersions, and says, how would be the co-crystallization part in the future of enhancing the properties of poorly classified drugs?
Sorry, Carmen, I couldn't hear the question.
How would be the co-crystallization in general? How would this take part enhancing the properties of poorly classified drugs? What is your take on co-crystallization? Because co-crystallization is also used in of course, when we need to bring a drug on the market, we try a polymorph screen. If it doesn't have the right solubility or dissolution profile, physical-chem properties, we go further and we investigate salt formation. And after salt formation, if you cannot make salts with that API, for example, you go through co-crystallization. And if also co-crystallization fails, at least my experience in pharma is that as a last resource, you would use amorphous solid dispersion. But taking one step before that, co-crystallization. Co-crystallization indeed is also used to enhance physical chemical properties of drugs. It highly depends on the API. It can definitely work for some, and some of them, at least also I have worked in my career with many drugs that you would not have any option with it. The only one would have been amorphous solid dispersions, but definitely co-crystallization is a big part. And they are, in the last 5 years, 10 years, I can even dare to say, a lot of publications out there where it is tried co-crystallization with another drug. So you actually tackle two pain points in the same time. It helps quite a lot. There are articles out there. I don't know, Sharmita, if you hear me, what is your opinion about co-crystallization in general?
Yeah, I mean, absolutely, Carmen, you are right. I mean, I am completely in agreement with that. That is definitely one of the another strategy for improving the solubility. So there are multiple strategies, and depending on the APIs, and the kind of increase of solubility that is required. Any of these techniques can be useful. It is very difficult to say that, one strategy is better than the other.
Yes.
It's a very system specific and project specific requirements that needs to be looked at.
And I believe big pharma companies, I do remember many years back, I think even 15 years back, they moved to strategies, I think, if I remember, yes, 15 years back, and they would explore all the possibilities, not only polymer, not only salt formation, co-crystallization. They would nevertheless would explore all possibilities that you can do with a API, also to be able to cover their IP, I guess. So you would always look at all strategies, co-crystallization, amorphous solid dispersion, everything to cover, if you want to cover your, from an IP point of view, of course. And definitely, if you are working for a generic company, you definitely have to cover all these and see what are maybe missing points that they were not covered, and think out of the box to try to cover all the landscape. Another question is, and I think is the last one that I'm going to take, because I see also the time is, we went quite a lot over the time, but I'm thankful for all these questions. We had around 30 questions, so it's nice to see the interest of our attendees. Is there a possibility that we can determine the amorphous solubility through Crystal16? Of course, if you have, I will take this one, Sharmita. If you have a stable amorphous dispersion, I would definitely try the Crystal16 to, and the added value of the Crystal16 is that you have you can do temperature dependent solubility. So you can easily define the solubility curve, and not only that, also the supersaturation curve and so. So it will definitely give you quite a lot of information. I will I will stop here. I see that the same person saying pertaining to API only, not ASD. Of course, API, not ASD, salts or crystals. What do you have? Everything is possible with solubility determination on the Crystal16. And the beauty of it, once again, that you have temperature and you can, in a controlled way, scan for, let's say, measure through a wide range of temperatures. It can give you the temperature dependent solubility. So you are looking for that cloud point, of course. I will try to stop here this webinar. We went quite a bit over it. I would like to thank you all for your patience today. We had a few technical problems. You will have the recording in the coming days, coming in on your inbox with an email, and of course also the slides from our presenters today. If you still have questions after this webinar, please feel free to contact us via info@crystallizationsystems.com, or you can send it directly to me, carmen.guguda@technobiz.com, or via the website, and we will do our best to answer your questions. Once again, thank you very much for your patience today, all of us. thank you, Sharmita and Jayshree, for your time and your patience and sharing such a great case study with all our attendees. The number of questions shows a very high interest, so well done, and thank you very much for sharing this. I'm looking forward for another